Hi everyone, this is Susan Rudiger and I am the founder and the CEO of the CMT Research Foundation. I'm really grateful that you all have joined our webinar today celebrating 1 year of business of the CMT Research Foundation. 1 year ago today, Pat Livney and I launched the CMT Research Foundation. Many of you on the phone here know that both Pat and I have CMT, and we want to move the needle on drug development for CMT to develop treatments and cures. So many of you know our leadership team, and the three of us are tonight's speakers. I obviously am going to start us off giving you a little bit of a picture of the CMT Research Foundation, and then Pat's going to come in and tell you about why we started the Research Foundation. Grace is going to go over the scientific side of things, talk about the drug development pipeline and where the CMT Research Foundation is making some inroads now and where we intend to move the needle moving forward. And then we'll have our Q&A session, and then we will wrap up with some closing slides. Hopefully, after this evening's presentation, you'll understand more about what it takes to get treatments delivered and approved for CMT, and how the Research Foundation is changing the drug development landscape. So I'd like to introduce Pat Livney to talk a little bit about why he and I started this endeavor.
As everyone on the phone, or most of y'all know, Susan and I spent a lot of time working on behalf of CMT and CMT patients at the CMTIA, certainly over 10 years each in our capacities, and we're really proud that we were able to move the needle the way we were, both on the research side as well as programs. At the end of 2016, I left. At the end of 2017, Susan left. And we weren't about to leave the commitment we had to finding a first treatment for CMT. So from a white paper and a discussion with Susan and in talking her into becoming a CEO, we started this endeavor just 12 months ago, a short 12 months ago. So my passion as a person with CMT, an axonal form, and Susan's passion as a patient with a demyelinating form. You have all the bases covered as to what our mission statement is.
Some of the reasons why Pat and I started the CMT Research Foundation start here. You know, Pat and I both have CMT, and we are really laser focused on getting treatments approved. We are frustrated by the pace of research, and some of the obstacles that we see in the landscape are these. Drug development is costly. Some estimates show us that it's, around $1 billion to deliver a drug from zero all the way through the approval. And that will include all of the failures that happen in drug development, that cost. And as we know, you start with a lot of compounds to begin with, your pipeline narrows down and you get one approved. So that $1 billion takes us all the way through the approval of one drug. That also talks about the second point, which is only 12% of drugs in clinical trials get approved. And that's a problem for rare diseases. But the good news is that of the 59 approved drugs in 2018, more than half of them were for rare diseases. We know that pharmaceutical companies, biotechs, FDA, NIH, they're focused on moving the needle for rare diseases. And for those of you who don't know, CMT is included as a rare disease. But none of the drugs that were approved in 2018, or ever for that matter, were for CMT. And Pat and I want to change that.
Susan knows me well enough to know that I like pictures, and pictures are kind of worth 1,000 words sometimes. We at the CMTRF don't really consider anything without cash flows and meaning, as well as a diagram to show exactly what we're doing. But this picture here tells you exactly where CMT is in the spectrum of drug development and treatments. And this picture really tells you why Susan and I, a dedicated board, a talented SAB, and our chief science officers are all dedicated to trying to change that needle into getting treatments to patients. I could tell you, if you look at MS, for instance, which is considered really the CMT of the central nervous system, you've got 17 plus treatments, and they're all every year they're getting better and better for MS. And you notice for CMT, we have 0 at this point. That's why we started this foundation, and quite frankly, there's a huge room to add additional capital and energy to CMT disease and getting us across the goal line on some of these treatments. I will tell you that our job is not just us in our little community at the CMTRF as Susan grows this organization, but it's incumbent on all of us. Anyone who has a touch point to this disease is responsible, and we feel responsible, and hopefully joins the efforts in raising capital. The hallmark of our foundation is to deploy that capital with prudence to high-impact projects. I can tell you Susan came from a bio meeting where 30 different companies are attacking her as to what we're about, what our mission statement is, and 30 companies that weren't involved just 5 years ago. So at the end of the day, when our process, which includes a 6-week approval process, which blows away anyone out there in the space, headed by Grace and our SAB, we look to lever our investments, accelerate the development for treatments. We do our projects in a milestone-based format, and we insist on sharing in the economics, should there be any, of the project that we are funding. So from our standpoint, we're unencumbered. We're not beholden to anyone. There are no politics. There is a single focus, and that's to find a treatment for CMT, regardless of letter or number associated with it, as fast as possible. So from the standpoint of our mission statement, no one ever has to worry about what Susan does 24/7. It's, does it hit our mission statement? Are we hitting the hallmarks of what we do and how we fund and how we operate? If so, go. If not, kill. And if there's something in between, there's a robust discussion to maybe cherry pick the best of the LOI that comes our way. So hopefully, with this short amount of time that I've got, and before I turn it over to Susan, I want to say a little bit about Susan and why she's the ideal person to head this organization. I tell her daily that she's one of the top 5 people of import in the CMT patient community as well as research. And what I mean by that, and she'll go through this with Grace, in a short 12 months, we become as big and plan on being the biggest in the universe, focused solely on research. Susan comes from a family of 5 generations of CMT-afflicted people. I am a de novo mutation and waiting to get confirmation on that mutation. We have a lot in common, so I couldn't be more proud of what Susan has accomplished in 12 months in building this organization and continue to build this organization. And with that, now I'm going to turn it over to a very, very talented lady.
So as the CEO of the CMT Research Foundation, I'm going to tell you a little bit about who we are as an organization. We have 3 key tenets of the organization, but one focus. One of our clear tenets is our clear strategy, and that is that we exist for one reason, and that's to deliver treatments for CMT. It is our only focus and the only way that we are going to deploy the funds from a program perspective. That being said, we are highly efficient with our dollars. We've learned how to stretch every dollar, increasing the impact of those donations made to the foundation. And I'm really grateful that we have Grace on board. She has deep expertise in vetting projects. She knows how to keep budgets honest because she's written them as an academic. And then she also knows how to parse them out because she's vetted millions of dollars worth of projects in her former life at the Muscular Dystrophy Association. The last key tenet, and this one is really important to both me and Pat, and that's transparency. We will be transparent with everything that we do.
The best thing we can do is make sure that every dollar that comes in knows exactly where it's going and how it's spent. From our standpoint, without giving away proprietary information from the standpoint of the project itself, everyone will have the exact same point of reference of where that project stands. So, from a strategic standpoint, from an efficiency standpoint and a transparency standpoint, put us up against any 501c3 out there. 1 year into this foundation, I can tell you that we committed over $1 million of the monies coming in. As of today, we've raised over $2.6 million, which is phenomenal. We have funded 3 projects. Grace is pouring over 6 proposals. Right now, I know she has nixed 4 that haven't fit the criteria that we've set, as well as the SAB. We're dealing with 25 different companies within that framework, and a lot of it is teamwork as well. So rest assured, between our SAB and the other experts out there, that we're in contact on these projects. And then obviously, being part of the IMC, we've got that contact naturally with the 300 investigators worldwide. So obviously, from the standpoint of our first hire, Grace has been a phenomenal addition and has eased the burden on the science part of the equation, and is incredibly talented in what she is doing and bringing to this organization.
Let's level the playing field a little bit and talk about CMT. Everybody on the phone here either has CMT or knows somebody who has CMT, and we know what it can do to the body, right? I don't have to go over that for you a little bit. But let's talk about some of the basics of CMT here so we can all have the same understanding about what we're talking about. So CMT affects 1 in 2,500 people around the world. And just so you know, that is the same global prevalence as multiple sclerosis. It's actually 10 times the global prevalence of ALS. So that's how common CMT is. CMT is caused by genetic mutations, so it's a genetic disease. I think everybody on the phone knows that, but did you know that there were 100 different genes right now that have been identified to cause CMT? The scientific community actually believes that there could be another 100 that are still undiscovered. And Pat mentioned we haven't found his gene yet. He's one of them. The good news is that about 90% of the people who have CMT have one of 4 types: CMT1A, CMT1B, 2A, and 1X. And so 90% of us have one of these 4 types. We also have 4 different types of CMT in the nomenclature that I want to make sure everyone understands. You've got the demyelinating types, which are CMT type 1. And if you take a look at this cartoon over here, you will see that the myelin is the insulation on top of the axon of the cell. And so if you have a CMT type 1, it's demyelinating, where this myelin or this insulation breaks down. If you have a CMT type 2, it's axonal. It's a disease of the axon, and that's what the myelin insulates. If you have a CMT type X, it means that it's X-linked and that it's carried on the X chromosome. And another one to point out that you'll read about is CMT type 4, which means that the inheritance is recessive. It means you have to have 2 copies of the gene in order to have the disease symptoms, not just 1 copy like the others. So with that being said, we all have this level playing field about understanding the different nomenclature in CMT. I'm going to turn it over to Grace to talk about the different therapies.
Great. Thanks so much, Susan. We're going to switch to the science part of the presentation. So what I'd like to do in this section is share with you the different types of therapies that are in development for CMT, and also discuss how the CMTRF is helping to de-risk the therapy development process at different stages along the pipeline. So if you look at any disease, therapies can fall into one of 2 categories. They're either treating the symptoms or they're actually addressing the underlying cause of the disease. This latter form is called disease modifying. And disease modifying treatments can either slow down or halt or even reverse or cure a disease. And at the CMTRF, we're very much interested in funding science that specifically leads to disease-modifying treatments. So let's look at the 3 general classes of therapies that are currently being developed for CMT in industry. First off, there are small molecules, and small molecules are chemicals that are synthesized in the laboratory, and they're usually administered to humans systemically. And depending on the specific drug, these could either be modify the symptoms or treat the symptoms, or they could be disease modifying. For example, small molecules could slow down or prevent the death of nerve cells, or they could help to provide more energy to the nerve cells to make them function better. So moving along to cell therapy, in this type of therapy, cells can be isolated from various types of human tissues. They're purified, and then they're injected locally near specific nerves. And the point here is to try to enhance the myelination and make the nerves conduct electrical impulses much better. So this approach would be considered disease modifying in very limited number of regions of the body wherever those cells were injected. So lastly, gene therapies there on the right, they target the underlying cause of disease. They either add, they remove, or they modulate genetic information in some way, either in nerve cells or in the Schwann cells. And gene therapies are delivered either through modified viruses, usually something called AAV, or synthetic nucleic acids called antisense oligonucleotides, or ASO for short. Now, these therapies can be administered either locally or systemically. One of the current challenges with these various gene therapies is that the peripheral nerves are not very efficient at taking up viruses or ASOs. And so one of the research priorities for the CMTRF is to fund projects that solve this delivery challenge of gene therapies to the peripheral nervous system. So I know that not everybody knows what the drug discovery and development processes looks like. So how do drugs go from the laboratory to your medicine cabinet? Every drug in this country has to travel the same road to final approval by the Food and Drug Administration, or the FDA. So on average, this is a 12-year process. Now let's take a very simplified look at what this process looks like in CMT. So there on the left is the discovery phase. First, scientists need to gain a better understanding of the underlying disease mechanism through either identifying the gene that is causing a specific type of CMT or identifying the pathways that are altered in nerves and Schwann cells and lead to the pathology seen in the disease. Then a target in the cell needs to be identified, which could be potentially modifiable by a drug. And so targets are generally genes or proteins. So next, going to the preclinical stage, scientists screen for drug candidates in models. And these models can be either cultured cells, typically adult stem cells, which are differentiated into nerve cells, or they're animal models, typically mice or rats. And a good candidate that comes out of this stage for future tests in humans will be able to modify the nerve pathology in animals and will not be toxic. So once the data is in hand by a company that's showing good efficacy early on, the company will submit the data to the FDA, and the FDA will analyze the data and evaluate whether the therapy can then move ahead to be tested in humans. So in a phase 1 clinical trial, this is the first test of this actual candidate in a human. And the candidate's usually tested at several doses, typically in a small group of people without CMT, but not always. And the purpose here is to determine whether the drug is safe for humans. If the safety profile looks good, then it's on to phase 2 clinical trials. Here the candidate's tested in a small cohort of people with CMT to determine if the drug is effective in causing beneficial changes that can be measured by the researchers. For example, do the nerves function better? Has the muscle strength changed? Can somebody walk faster or further? If these are working there, then in phase 3, further tests are done in a larger group of patients, again, measuring if the drug is causing beneficial changes. If the data does show a beneficial change in the disease, then the company submits a new drug application to the FDA for review, and then FDA experts evaluate the data and determine whether the drug is approved. Now let's look at the 16 companies that are currently at different phases of drug development for CMT. This slide shows tremendous progress. As 5 years ago, if we drew this slide, there wouldn't have been anything on it. So there is definitely activity starting to happen in the CMT space. There's some general take-home messages that I want you to walk away from here. So one of these is that the majority of therapies that are under development in the industry are for the 1A subtype, with the second most common being 2A. And this is due to both the underlying biology and the ability to target that biology, as well as the market size. These are among the most common forms of CMT. Another take-home message is that most of the therapies here in development are either small molecules in green or gene therapy in blue. Furthermore, the therapies can be found throughout the development process, from the earliest discovery stage to one of them in phase 3 clinical trials. And the longer the bar, that indicates the further along that particular compound is in the process of drug development. And if you'll notice, most of these are at very early stages, and there's only one in phase 3 right now, and that's a combination of 3 different drugs that are already approved, so not surprising that that's the furthest along. Another thing to glean from this slide is that these drugs that are under development are targeting a variety of different pathways in the nerve cells or the myelin-producing cells, and most have the potential to be disease-modifying. We here at the CMTRF are staying abreast of this pipeline, and we're in regular contact with many of the companies that are listed here. We support their work, and we look for ways to complement this pipeline through what we're doing at the foundation. Additionally, we're working to solve some of the obstacles that some of these drugs face to get approval. So on the next slide, let's look to see how the CMTRF is working to de-risk the drug development process at multiple points along the pathway. So we know that therapy development is never an easy road for any disease. CMT presents its own challenges in this regard, but CMTRF is addressing these challenges in multiple ways. We'll start at the bottom of the slide at the 2 items in yellow there. So CMT is a complex disease. There's over 100 causative genes currently known. However, there are patients who still don't have a causative gene identified, like Pat. In addition, while researchers know the primary targets, for example, for 1A, many targets in other subtypes are still not identified. And therefore, it's really hard, if you don't have that target, to test which drugs might be beneficial. So at the CMTRF, we prioritize initiatives that can have application to multiple subtypes. This is nicely illustrated by 2 of the projects we're funding. For example, we're funding a drug screening project in 2A, which could be beneficial for multiple subtypes, which are primarily axonal in nature. The gene silencing approach that we're funding in 1A could be applied to other forms of CMT. And I'll be talking more about these specific projects in a little bit more depth very shortly. So let's move to the middle of the slide to the 2 items in light green. So we know that testing drug candidates in cell and animal models is a necessary part of the drug development process. I described this earlier. So what we are doing here is we're prioritizing projects which use patient-derived stem cells or that lead to the creation of better animal models in which drugs can be tested. In addition, our scientific experts on our scientific advisory board carefully evaluate the projects that are coming to us to make sure that the animal and cell models that are being used in these projects are very appropriate for the studies. We also know that the drug development process is long and fraught with drugs failing at the safety and efficacy phases in clinical trials, so those phase 1 and phase 2 trials. So we aim, what we're trying to do here is we aim to shorten the timeline to drug development, and where possible, we fund projects that are using repurposed drugs, and these are drugs that have previously been tested in humans for other indications. And we are also using assays on patient cells, which are genetically authentic and will also help move things quicker through the pipeline. Finally, let's move to the top of the slide to the 2 items in dark green. CMT is a slowly progressive disease, and in slowly progressive disease, it makes it much more difficult to determine in a clinical trial if the therapy that you're testing, if it's improving the disease. Because if you don't see very much change over a certain time, then it's very hard to know if your drug is having an effect. And you can't do a clinical trial for 3 years, very expensive. So what we're doing in this regard, we're working with key leaders in the field to support clinical trial readiness efforts and make sure that the best outcome measures are being used where appropriate in clinical trials. Finally, drug development for CMT is costly, and there's not enough money to fund these efforts. So what we do is we carefully evaluate projects to make sure that we're investing our money in the best projects in academia and industry that best de-risk and expedite therapy development at critical stages of the process. We also have a vested interest in the success of each project. Should the project advance to commercial stages, the CMTRF would be included in any financial success, which will allow us to put more funds back and reinvest into research to help drive therapy development even further. So now I'm very excited to share updates on the 3 projects that we are currently funding. So let's look at the first one, which is a drug screen, and the goal of it is to identify candidates that can halt axonal degeneration in the 2A subtype. And so this project is being funded in industry to a company called CuraStem. So what this company is doing is screening 2,800 repurposed drugs on patient-derived stem cells, which have been differentiated into neurons in the laboratory. And this type of assay is beneficial for 2 reasons. The first of these, as I already kind of mentioned with the repurposed drugs, is that repurposed drugs have already been tested in people for other diseases, so there's a faster path to drug approval in CMT. And then second reason here is that patient-derived stem cells are, of course, human in origin, so they have all the correct genetic material. And so that increases the likelihood of success and translation of these candidates into clinical trials. So the company just presented their first 6-month results to Susan and I, and we were really pleased to hear that they already have 1 initial candidate that slows down cell death in the cultured patient cells. They are now going to move on to the next phase of the project and screen that full complement of 2,800 drugs in order to find better candidates that are able to increase the survival of these cells in the culture dish. The candidates that are identified from the screen could potentially move right into phase 2 clinical trials in people with CMT because these are a cohort of repurposed drugs. The other interesting thing about this project is that this assay platform that they're using to screen the drugs and determine if the cells are living longer in culture is that this type of approach could be applied to drug screening for multiple types of CMT using patient-derived stem cells from individuals with other subtypes of CMT. So stay tuned. They will have more updates on this project in the future. So with that, let's move to the second project that we're funding, which is a gene therapy approach in CMT1A. And so this project is focused on developing a viral-mediated gene therapy for 1A, which is the most common subtype. So we know in 1A that excessive amounts of a protein called PMP22 are produced in Schwann cells. And Dr. Cleopa, who's a leader in gene therapy approaches for CMT, is developing what's called a gene silencing approach to reduce these excessive amounts of PMP22 in Schwann cells. So he's using experimental mice which have the human version of the PMP22 so that this silencing tool that he's developing has a greater chance of eventually translating to humans. He also presented his 6-month results to Susan and I recently, and what he's found is that the method that he's testing in these mice can indeed reduce the levels of PMP2 to some degree. So now in the next phase of the project, he's going to work to optimize this method to get better delivery of the virus to the Schwann cells, and then he'll test if the nerve disease is altered, is lessened or reversed in the experimental mice. So this type of gene therapy is very exciting for CMT. It has potential to be a one-time treatment for 1A, and that's what's called in the field as one and done. So you would only need to potentially administer it once, and you wouldn't have to give it again. So furthermore, the AAV virus approach has a higher potential to be approved for CMT, as it's been recently approved for use in other diseases. Finally, there's a lot of interest in industry to commercialize gene therapy approaches for CMT. In general, gene therapy approaches, a number of other diseases, there's a lot of interest also, so that's coming over to CMT also. So in the meantime, the CMTRF is committed to solving the problem of delivery of genetic either viruses or the ASOs to the peripheral nervous system. We're currently vetting 2 industrial projects which are aimed at enhancing delivery of these genetic techniques to Schwann cells. Those are currently being vetted by our scientific advisory board. So I'm going to move on to the third and final project that we're funding. It's the newest of the ones that we're funding, and it's targeting CMT1B, and it is to a leader in the 1B space called Dr. D'Antonio, and he's located in Italy. And this project is important because it's seeking to determine whether a pathologic pathway, which is called the unfolded protein response, and which is found in CMT1B, whether this pathway could be targeted by drugs after a patient has already had symptoms for a number of years. And the goal is to be able to stabilize or reverse axon degeneration in individuals who have been having symptoms for quite a while. So what this project does is using genetic methods in mouse models of CMT1B to provide what's known as a genetic proof of concept for this approach. And so if this approach is successful in this project, then there's a small molecule called SEPN1, which is currently in clinical trials for CMT1B, and it may then be able to be tested, and it may be beneficial for patients who have suffered with symptoms for decades. As I mentioned, that just started, so we won't have any updates on that for probably about 6 months or so. So I'll end my section by discussing how the progress that's been seen in drug approvals for other rare neuromuscular diseases really gives hope to the CMT community that these same type of disease-modifying approaches will be beneficial for CMT. Indeed, the projects that I just described, the 3 that we're funding, are using approaches like these drugs on the current slide. So 2 genetic therapies have been approved for spinal muscular atrophy, one in 2015 and one just earlier this year. So Spinraza is an antisense oligonucleotide, or an ASO, as I used that term before. And what this drug does is target genetic instructions in nerve cells and increases the production of the SMN protein, which is needed for nerve survival. It's administered 3 to 4 times a year for the life of a patient. And this type of technology, this ASO technology, is being tested currently in CMTA, CMT1A, and we're currently vetting 2 biotech companies who've submitted projects for consideration of ASO therapy, and we'll be reviewing that by the Scientific Advisory Board. So Zolgensma is the second spinal muscular atrophy drug that was just approved this year, and in this drug is a viral vector called AAV9, and it's delivering a normal copy of the SMN gene to nerves, and this is a one and done or a one-time treatment for this particular virus. So this type of technology is the same that I described for the 1A project that we're funding for Dr. Cleopa. And finally there on the right, Keveyis is an example of a disease-modifying small molecule. This drug was approved for neuromuscular disease called periodic paralysis in 2015, and it changes the levels of potassium in nerve cells so that the nerve is now able to function properly and individuals don't have those periodic episodes of paralysis, hence the name of the disease. And as I mentioned, this is a disease-modifying small molecule, and we are funding, the project that we're funding to CuraStem is a small molecule approach to prevent nerve cell death in 2A, and this also has the potential to be disease-modifying. So with that, Susan, I will throw the floor back to you.
Oh, thank you, Grace. So we actually have a couple minutes here that we can ask some questions from our panel. I have received a couple questions coming through Grace's presentation. And one of the questions that came through is a question that I seem to get a lot from the patient community, which is there are so many efforts aimed at CMT1A. What hope do those of us with rare types of CMT have? So, you know, we mentioned earlier that CMT1A is the most common type. And for CMT1A, everybody who has it, which is over 50% of people who have CMT, everyone who has it has the exact same genetic mutation. We have, and I say we because it's the kind that I have, we have a duplication of a gene that creates an overexpression of a protein. And we understand the gene that causes it, and then we also understand, for the most part, the target. We want to downregulate that overexpression of the protein. So because we know the gene and because we know the target, that makes it really viable for drug development. But then the really important part, or another important part, can be that commercial viability. You know, if 3 million people in the world have CMT, that means that 1.5 of us have CMT1A. So it makes it commercially attractive to the pharmaceutical companies. But, you know, for the rare types, most of the rare types are type 2, and type 2 are axonal. And so a number of the projects that are out there right now talk about the axon degeneration or targeting axon regeneration. And especially if we take the small molecule approach to this, we're pretty sure that some of the therapies that are going to work for maybe, let's say, a pharmaceutical company is looking at 2A because of its prevalence. If it regenerates an axon, it may have the potential of being applicable to everybody who has a degenerating axonal variety of CMT. So we think that those people with the rare types, the genetic approach won't be immediate for you, but we do think that there are small molecules out there that will have a broad applicability to a number of different types of CMTs.
Mm-hmm.
Do you want to add anything to that, Grace?
Sure. So this is where, like, understanding, there's been a lot of work done on the basic biology of axons and nerves and how nerve cells die. And some of these pathways are well known, and so some of the drugs that are being tested, or pathways that are being tested as targets, are targeting that very latest step by which a cell dies. So again, that could be applicable across multiple of the axonal types. So that's a specific example of something in that realm.
Excellent, thank you. So another question that I have here is gene therapy the only way that we can cure CMT?
No, that was where, like, the small molecules, that some of those have the potential to be disease modifying. They can, as I said, prevent the axonal death. There could be possibly, the one that's targeting the unfolded protein response in some forms of 1B, that could also be a beneficial small molecule. So there are definitely potential for small molecules to be disease modifying and, quote, curative.
Thank you. We got a question here that says, you know, there seem to be an awful lot of drugs in the pipeline. Do we really need more drugs in the pipeline? What did you say, there were 14 different drugs, Grace?
There's 16, various stages, a lot of them at the early stage. There's a lot of drugs don't make it. And so you want to have multiple shots on goal. It's possible that all 16 of those drugs that I showed will fail, or they're years away. So you don't want to put all your eggs in one basket. You definitely want to be making sure. And plus, there may be new pathways that we haven't even targeted that are even better. And also, even if any of these drugs did come to approval, there's always improvements that can happen, and there's many examples where the first generation drug comes out. It's not too, it's okay, but that second and third generation follow-on drugs are even better. So even if we got something today, that may not be the end all and be all.
We have a question here that says, have we run across anyone who is using approved drugs off-label to treat forms of CMT with any success? I don't have any experience with any or any knowledge about any off-label drugs. Grace, do you have any knowledge about that?
No, I don't. As I mentioned, the Pharnex it's not off-label, but that is a test of 3 different approved drugs being used in a cocktail. And that one is in phase 3, but they were just told by the FDA that they needed to go back and do more studies, that there was problems with the data. So I'm not sure that really counts as an off-label, but at least those are some things that are already out there, which I think was the intent of the question.
Yeah, okay. And we have one more question from Jack, and I'm going to get to your question, Jack, because you asked a question that I will answer here. But for those of you who didn't have your questions answered, I want to make sure that you have the contact information for all 3 of us. So feel free to email us anytime. If you want to set up a phone call to talk with us, our phones are always available. We're always available to talk to you. I started off the presentation by saying that transparency is a key tenet of ours, and we welcome a dialogue with every one of you on the phone. So start off by sending an email with your questions, and we'll be more than happy to talk to you and get your questions answered. So thank you for those of you who submitted them. So I want to show you what the future of CMT looks like, and I want to do this at the same time as answering Jack's question. You know that we're leveraging the existing knowledge about drug development and applying it to CMT. We're very focused on our efforts, which can translate into wins for the CMT community. And you'll remember that we have these 6 more proposals that we're currently vetting, and we'll have some announcements about those soon. But you probably all want to know, okay, so what do you 3 envision the future of CMT to look like? And Jack, this is where your question comes in, because Pat showed this chart to you at the beginning of the call. And Jack wanted to know, why does the chart look like this? Why are there so many trials done for ALS? Why are there treatments available for all of the other diseases except for CMT? What is the problem here? And, what can be in store for CMT? I would like to see the blue DNA line, which is the number of trials completed. I'd like to see that match ALS or MS. And certainly the number of treatments available, we want to see that number, that bar graph go off the page as well. That's what we think the future of CMT looks like. But how do we move that forward? That's the question that everybody asks. Why do we lag, and how do we move it forward? And the answer lies in patient-led research efforts. And I want to give you a couple different examples about this. Let's start with Duchenne muscular dystrophy, which is the second one there. So Duchenne muscular dystrophy has a small patient-led group called Cure Duchenne. It was started by 2 parents who had a son who had Duchenne muscular dystrophy. They raised $35 million, very smartly invested it in pharma, biotech, as well as a few academic projects. And their funding led to 12 clinical trials. And the clinical trials led to the first FDA-approved therapy. Cure Duchenne is laser focused on advancing the research for Duchenne. They raised the capital to get it done, they deployed it properly, and they have an approved treatment, or there's an approved treatment because of their efforts. So they set out to find treatments for Duchenne, and they succeeded. Another example that we can find here is spinal muscular atrophy. There's an organization out there called the SMA Foundation, and they were also started by parents of a child who had SMA. So 12 years ago, they opened their doors. They've raised $150 million, again, invested it wisely in both academic and pharma And now there are 2 approved treatments, which Grace talked about for SMA. And the SMA Foundation was really instrumental in getting those across the goal line. And if you want to know the best news of all, and this I aspire to, the SMA Foundation, they're now closing their doors as a foundation because they did what they set out to do. They had a mission, they delivered on it, and they're closing. They're moving on. And that, I think, is really admirable. So it's the concerted efforts by the patients who are driven to advance these therapies for rare diseases. And at the Research Foundation, we understand this urgency and we share it with you all. You know, Pat, Grace, and I, there's no time to waste, there's no money to waste. We need to bring it in, we need to deploy it smartly, and we need to reach some major milestones. I personally, you've heard, I come from 5 generations of CMT, and all of us have really seen the devastation that it causes. You know, I remember my great-grandmother in her wheelchair, and of course my grandmother, my mom, my aunt, my uncle, my sister, my cousins. You know, there are 5 generations of us, and I'm now seeing my cousins' kids who have CMT. And I don't know about you guys, but I feel like 5 generations of CMT is way too many. I really want this one to be the last generation that we have. This is why Pat and I started the Research Foundation, because CMT deserves patient-led efforts that have the single focus on advancing research. It's why we're here, and CMT deserves it. So the Research Foundation is the only organization with that single laser focus and the drive. We're fueling the research that leads to a funding where this graph becomes this graph. You know, but Grace, Pat, and I, the board of directors, the scientific advisory board, we can't do it alone. We're a small team and we cannot do it alone. And really, we need your help. Actually, quite frankly, we need your investment. On the line tonight, we've got a number of people who are significant donors to the Research Foundation. You all were big believers in us from the outset. You know that the early money is the important money, and you also share that sense of urgency. And we're really, really grateful for you. Thank you so much for letting us get to the $2.6 million in 12 months. It's delightful. What you hear a little bit of leap in my voice, because today we got a commitment for $200,000 from one person, and yesterday we got a commitment for $500,000 from one person. It's the largest gift that that family has ever given to the CMT space, or I think any charitable gift at all. This is really exciting stuff. We're building momentum. We've got it, but we can't stop there. You know, 2.6 million isn't enough. You heard Cure Duchenne was 35 million. SMA was 150 million. We need to have those kinds of numbers to get to treatments and to move this chart and to look like this. An important component of that is not just stopping with you and your investment, but getting your friends and your family involved. I can tell you that every single board member has made a donation themselves, but they've also inspired their friends and their families to give. We also see that from our donor community. These aren't just touch points of people who have the disease. They're friends of people who have the disease who say, I want to see this person live a healthier and happier lifestyle. And I have the ability to get that done. I have the ability to help accelerate that. So that's really what we need, is we need everyone invested into the Research Foundation, invested into our vision of a cure for CMT, and invested in ending this legacy. I know you guys are invested because it's why you took the time on a Thursday night to be here with us tonight. You are invested in our outcomes, and you're invested in the progress that we make. I hope that instead of just hoping and praying that people, the right people, will be moving the right money along, that you actually become a part of the solution and that you join us. It's going to be what it takes to end this legacy of CMT, not only for ourselves, but for the 3 million people around the world who have it. So I hope that we've answered your questions tonight. I hope that we've given you what you need. I hope that you are inspired to join us, and I hope that you'll contact us with any of your questions that you have. Thank you for everything that you all have done. Thank you for making these first 12 months a wonderful success. Thank you for giving us the ability to start these 3 projects and to take risks and to move the needle. I'm so proud of the work that we all have done together as a community, and I hope that you'll continue to support us and continue to be on this journey with us as we make this chart a reality for CMT. Thank you guys for joining us tonight. Have a great evening.